The entire surface of the Earth is covered with bacteria and other microorganisms, fungi and viral particles. And apparently there's bacteria even way down into the Earth, about 1 kilometre down apparently there's still bacteria everywhere. And this is quite a nice natural environment, but other environments aren't as clean as this. Maybe you work in some that aren't quite as clean as this. But in any environment, there's bacteria everywhere. And yet, inside my tissues, it's sterile. I'm pleased to tell you, as far as I know, there's no bacteria at all inside my thumb. In fact, I'm sure there's not, otherwise I'd probably feel ill. And I've no obvious wounds or inflammatory areas there. So it's sterile inside the human body. It's quite amazing that we have this milieu of bacteria outside, and yet sterile internal environments. How is this achieved? Well, there's two main things to think about really. One is the immune system, and the other is the lymphatic system. These two are fairly closely tied together. So the lymphatic system is absolutely vital for this. Now, it's useful, I think, to think of the lymphatic system in two components. The way I think about it. There are the lymphatic capillaries and the lymphatic drainage vessels, the ducts, and I would include in that the lymph nodes as well. So lymphatic fluid is going to go from the lymphatic capillaries into the afferent lymphatic vessels, through the lymph nodes into the efferent lymphatic vessels before it gets back to the blood. So there's that whole system of the lymphatic vessels. But then also in the body there's lymphoid tissue. And lymphoid tissue is tissue which performs an immunological function. So we might think of the primary lymphatic organs like the bone marrow and the thymus gland, where the leukocytes, the white blood cells, the protective cells, become immunocompetent. And there's also other areas of lymphoid tissue throughout the body that are sometimes called secondary areas of lymphatic or lymphoid tissue. So you might think of the tonsils, the appendix has got a lot of lymphatic tissue. There's lymphatic nodules in various organs, and of course the spleen has got a lot of lymphatic tissue in it as well, performing this vital immunological function. So the lymph ducts really are the flow of the lymphatic fluid, and the lymphoid tissue is other immunological areas of the body where these immunological cells, particularly the lymphocytes, live. It's their habitat, where they're able to recognize any antigenic challenge, and above all where they're able to respond to that antigenic challenge. So let's start our study of the lymphatic system now by looking at the formation of the tissue fluid and the lymphatic fluid, how that's drained through the lymph nodes, and how that relates to the aim of whole body immunity. So we want to think about this journey of lymphatic fluid through the body. And we already know that there are numerous arterioles that break down into huge numbers of blood capillaries in the body. Millions of capillaries, blood going in from an arteriole, blood draining out via a venule. And the fluid in the arterial end of the capillary is at higher pressure. So tissue fluid is formed at the arterial end of the capillary. And that's good because we have tissue fluid, because here we have the tissue cells that need tissue fluid. So they're bathed in this physiological tissue fluid, which is excellent. And there's large proteins in the plasma. So the proteins, albumin especially, in the plasma generate an osmotic pressure. The proportion of the osmotic pressure generated by plasma proteins is called oncotic pressure. So that causes the reabsorption of the tissue fluid at the venous end. So the tissue fluid is formed at the arterial end of the capillary because the hydrostatic pressure, the blood pressure, is greater than the osmotic pressure, but the tissue fluid is reabsorbed at the venous end because the osmotic pressure is now greater than the hydrostatic pressure, so there's more sucking in. And most tissue fluid is reabsorbed like this, but not all of it. Some tissue fluid is left in the tissues. And as well as this, sometimes plasma proteins escape. They get out of the blood vessels where they're supposed to be contained and they escape. Now, if we have plasma proteins that have escaped, these are going to be very osmotic molecules, and that's going to suck water. They're going to attract water to them. So if we have a lot of proteins in the tissue spaces, then that's going to lead to edema. It's going to be swelling in the tissue spaces, and that's bad for all sorts of reasons. It increases the diffusional distance that the oxygen has to travel, for example, or the distance that the carbon dioxide has to travel to get from the cells back into the blood. We don't want this excessive tissue fluid. So we have to get rid of some tissue fluid, and importantly we have to get rid of any proteins that escape as well. Absolutely vital that these are taken away from the tissue spaces. And as well as that, sometimes bacteria get into the tissue spaces. Maybe a cut, maybe a burn, maybe an infection. And bacteria left on their own devices can double in number every 20 minutes or half an hour. So we don't want to leave those in the tissue spaces for too long, because we're going to get a rip-roaring infection. So these all need to be taken away from the tissue spaces. Unfortunately, we have a system that does exactly that. So in between the cells in the tissues here, we have these blind-ended lymphatic capillaries like this. So finger-like projections, blind-ended lymphatic capillaries like this. And these drain away to form larger lymphatic vessels, slightly larger lymphatic vessels. And what happens is the excess tissue fluid is drained into these blind-ended lymphatic capillaries. And from there it drains away like this. And these lymphatic vessels here that we've drawn in green, the larger ones, so there's another set of capillaries there. There's a whole system of these. And these larger ones have valves to ensure one-way flow of the lymphatic fluid away from the tissues. So I would say that any fluid that is in the tissue is a tissue fluid, but once it crosses over into the lymphatic capillary, then I would then define that as lymphatic fluid. It becomes lymphatic fluid once it gets into this lymphatic drainage system. And as well as that, the escaped proteins will go into the lymphatic capillaries, because remember, we don't want those hanging around, and they too will be drained away. And as the escape proteins are drained away, that's going to lower the osmotic pressure of the tissue spaces, meaning that the edema will not develop, because there's no longer these large protein molecules to suck the water to themselves to increase the volume of fluid in the tissue spaces, because the protein has been taken away. And if there's any infection, then bacterial cells will also go into these lymphatic capillaries and also be drained away from the tissues. This is an important way that the tissues are kept sterile. Now, fortunately, the cells that line the blood capillaries, the bacteria normally can't get into those. So if there's bacteria in the tissues, that doesn't get into the blood circulation, because that will cause an instant bacteremia. But the capillaries can easily get into the lymphatic capillaries. So the bacteria can't get into the blood capillaries, but they can get into the lymphatic capillaries. And it's just as well as this way around, because if it wasn't, any minor tissue infection would cause a bacteremia, with of course the great potential for sepsis developing in that situation. So these are taking them all away like this. Now the lymphatic capillaries themselves, that we notice they're blind-ended, So there's only the flow of lymphatic fluid away from the tissue spaces towards the more central lymphatics. But the lymphatic capillaries themselves are made up of endothelial cells that overlap each other like this. They're flappy overlapping cells forming the lymphatic capillary. This would be the other side of it. So these flappy cells like this forming the lymphatic capillary. And what this means is, it's great because it means that the proteins, so it was a protein, the proteins can get through because these flaps flap in the way. They flap in the way. So increased pressure here and the proteins here will cause these cells to flap in the way, meaning the protein can get in because it flaps open. Same for the excess in tissue fluid. That can get in as well because these cells will flap in the way like this. So that will flap open like that to allow material in this case allowing excess tissue fluid in. But then when the fluids and the proteins are in the lymphatic capillary, that will slam the door shut. It's just like a door really, a one-way door. So that these are all like valves allowing this one-way flow. And it's the same if there's any bacteria as well. The bacteria can get in by this flapping open, but then it will shut to keep the material in and therefore to be drained away in the lymphatic system. So there's going to be movement, there's going to be gravity, there's going to be contraction of adjacent muscles, and this will increase the pressure in one length of the lymphatic system like this. So we notice that the valves open to allow material from there to there, but then these valves will shut to prevent retrograde flow of material, so it can only go in that one direction, so it's taken away from the tissue spaces. And as well as that, there's actually radial anchoring filaments on some of the lymphatic cells like this, the endothelial cells that comprise the lymphatic capillary. These are radial anchoring filaments because they're round about the capillary like this. And these are made of strong tissue and they're also elastic, connective tissue and elastic tissue. And these are actually anchored into the collagen fibres that are found in the tissues. Most tissues have some collagen, structural collagen within them. So these will keep the lymphatic capillaries open, facilitating a patent lumen for the flow of the lymphatic fluid through the lymphatic capillaries into these larger lymphatic vessels. Now, I think I'll just show you just a bit of context, just a different diagram here really. This is from my physiology notes book. So here we have blood coming in at the arterial end, going through the blood capillary, giving up its oxygen, leaving us darker red blood, like this. Here we have the tissue cells, so these are the tissue cells here that comprise the specific tissue that we're in. Could be any tissue of the body basically. The tissue cells. As we've noted, some tissue fluid can escape, some proteins can escape, and they're going to go into the lymphatic capillary. So let's just, in this case we'll colour the lymphatic capillary in yellow. We can see now the fluid is getting in from the tissue spaces into these blind-ended lymphatic capillaries. It's not getting out, it's a one-way system. And this is then drained away into progressively larger vessels. And of course, here was the lymphatic vessels draining to form progressively larger lymphatic vessels. And here we see just an individual cell, or the individual cells that make up the capillary. So this is the lumen of the lymphatic capillary. These are the endothelial cells, and as we've noticed, these are floppy to allow material in. So they'll flop open to allow material in, but then they won't let the material out again. The doors will shut to stop it going out. Now, just to lead us on to the next part of this account, we notice that these vessels, these larger lymphatic vessels, taking lymphatic fluid away from the tissues, these are described as afferent lymphatic vessels. So these are afferent, as we saw there, afferent lymphatic vessels. And an afferent lymphatic vessel is any vessel which is going to be taking lymphatic fluid towards a lymph node. So afferent vessels, so this is an afferent vessel because this is going to be carrying material towards a lymph node. So next we need to think about what is happening with these lymph nodes.